The interesting thing about homologs is that they're going to separate independently during meiosis 1. All possible combinations of alleles are going to occur in the gametes. So if we were looking at a particular dihybrid cross, meaning that we have an individual, so in this case we're looking at individuals with multiple traits. We're going to be looking at individuals who either have freckles or don't have freckles and have short fingers versus long fingers. So here we have potentially an allele for finger length, or actually here is an allele for freckles versus non-freckles, and we have finger length, short finger length versus long finger length. Now the interesting thing is, when this individual first divides, they can separate their alleles in any number of ways. We could separate with short fingers and freckles, and long fingers and no freckles, or we could separate short fingers and no freckles, or long fingers and freckles. We can assort themselves any sort of way. So what we end up with is potentially we have an individual who is dominant for freckles and dominant for short fingers. Or we could end up with an individual who is recessive for freckles and recessive for short fingers. Or they could be recessive for freckles and dominant for short fingers. Or they could be dominant for dominant for freckles and recessive for short fingers. So in other words, just because an individual is dominant for one trait doesn't mean they're going to be dominant for all traits. Just because they have the dominant trait freckles doesn't mean they're going to have the dominant trait for short fingers. So here's an example of kind of what that looks like. This is a dihybrid cross. So a dihybrid cross is going to involve two traits, meaning that a person is going to be that if we start off with a person who is homozygous for freckles and short fingers, so homozygous dominant for freckles, homozygous dominant for short fingers, and they reproduce with an individual who has no freckles and long fingers, or they're homozygous recessive for both traits, what we're going to end up with is an entire generation of individuals who are going to be dominant for freckles and dominant for short fingers. However, if we were to cross individuals who were heterozygous for all of those traits, so we were to cross an individual who is heterozygous for freckles and heterozygous for short fingers, what we're going to end up with is a very interesting mix. We're going to end up with 9 individuals who have freckles and short fingers. So in other words, 9 individuals who are dominant for both traits. 3 individuals who are dominant for freckles, but recessive for finger length. 3 individuals who are recessive for freckles, but dominant for finger length. And 1 individual who is recessive for both freckles and both finger length. So what we're going to end up with is a 9-to-3 to 3-to-1 ratio. 9 who are going to be dominant for, or not 9 who are going to have the dominant phenotype in both cases. 1 that is going to have a dominant phenotype with a recessive phenotype. 3 with a dominant phenotype and recessive phenotype. 3 with a recessive phenotype and dominant phenotype, and 1 individual is going to be recessive for both traits. So a person with a dominant phenotype for 2 alleles could be homozygous dominant or heterozygous. But if an individual who is homozygous dominant reproduces with a homozygous recessive, all of the children are going to be completely dominant. If the individual is going to be dominant, if the individual is heterozygous, they're still going to show the dominant phenotype. So whether we are homozygous dominant or heterozygous, we're going to still see the dominant phenotype. However, when they reproduce with an individual who is homozygous recessive, each child will have a 25% chance of showing one or both of those recessive traits. So here we're going to, so for this section, you should be able to explain how the results of a dihybrid cross are related to the events of meiosis. In other words, bring this back to independent assortment and the fact that just because you're dominant for one trait doesn't mean that you're going to be dominant for all traits. But those traits assort independently during the stage of meiosis to introduce genetic variability. You should also be able to predict what genotype the children will have if one parent is homozygous recessive and for no freckles and homozygous dominant for short fingers, and the other parent is homozygous dominant for freckles and homozygous recessive for long fingers. And then you should be able to use a dihybrid cross as an example to explain how the gametes are formed by the process of meiosis. Now, I know this is a lot to kind of take in, and we don't spend a whole lot of time right now going over Punnett squares, but I promise they're a very important part of this section, and we will spend a lot of time practicing them in our live discussion time. So if you can make it, great. If you can't, but you would still like some extra help, please let me know. Send me a message. I'll be happy to talk through anything with you over email, or set up an extra time that you're available that we can meet and go over Punnett squares. All right.